Okay. All right, everyone. Good afternoon. My name is Claudia Regal. I'm the director of the City of New Orleans Mosquito, Termite, and Rodent Control Board. And for many of you, I think you may know that we host a webinar series. And today we have a good one. So today it is my pleasure to introduce you to Dr. Drs Carlson as well as Dr. Mark Janowiecki. And this is going to be a really interesting webinar from really a patient and physician perspective about alpha-gal syndrome. So Dr. Mark Janowiecki, he's our research entomologist. He's been a research entomologist here at the City of New Orleans for a few years. He's started in 2020. He's always had an interest in insects and studied termites through his several degrees in entomology, applying molecular tools. Differentiation. He currently leads the termite division for the City of New Orleans, which protects our facilities and historic buildings and trees from termites, but he also manages the molecular laboratory, which conducts pathogen surveillance in mosquitoes, rodents, rodent ectoparasites. We also do genetic research, screening of resistance, and that includes with mosquitoes and rodents as well. And so also Dr. Drs Carlson, he has received his medical degree from Tulane University, and also very interestingly enough, he has a BS in entomology from the University of Maryland, and he has a PhD in parasitology. He's a board certified American, he's board certified by the American Board of Allergy and Immunology. Dr. Carlson performs pediatric allergy and immunology. He also sees patients at the Ochsner Center for Primary Care and Wellness. So with that, I think Mark's going to be starting off the webinar, and thanks everyone for joining. Please, if you have any questions, please put that in the chat. We will be following it through. And so we're very excited to have all of you here today. And also, if you want to learn more, you can see the little logo at the bottom of the Gulf South Vector. I put the website in there because there's going to be a lot of future interesting webinars coming. So with that, Mark, please take it away.
Okay, awesome. Yeah, thank you. So I'm going to start off with a little bit of kind of tick biology and background, and then pass it to Dr. Carlson, who's going to introduce alpha-gal syndrome, and then I'll come back to me and talk about kind of my experience as a patient. So first off, for tick biology, so this is the part, hopefully if you understand enough, you can avoid getting this condition as well. So ticks, as we probably know, are small arachnids. They're not actually insects. The adults and nymphs have 8 legs, but the larvae do have 6. So this can be slightly confusing if you think about arachnids having 8 legs. The younger ones only have 6 when they're very small. And they find hosts by questing. Questing is when they climb up a blade of grass or some kind of material and try to find a host just holding their legs out. As you see this bottom picture, the tick has its front legs out, hoping something brushes up against it and it can grab on. So they're not jumping out of trees. They can't jump. They can't fly. There's many myths about, wearing a hat out to avoid them falling from the trees into your hair, but they're really usually coming up more from the substrate. For tick bite prevention, know where you can encounter ticks. So oftentimes being outside, when you're gardening, camping, hiking, playing outside. So anytime that you're outdoors in kind of these grassy areas, it's likely to encounter them. And when you're in these areas, use a repellent. So use something like DEET or permethrin treated clothes. There's clothing that you can get that has the permethrin already treated into it, and it's good for a certain number of washes. This is a great way to keep the ticks from crawling up to you. This is just going to keep them off when they get onto your clothing. For your outfit choice, light colors with long sleeves, socks, and long pants tucked into the socks is a good idea. So you can see the socks are tucked in here. This gives them less access points, and you're more likely to see those ticks crawling up your leg and be able to brush them off of you and not have them get into your clothing. And for our pets, use tick medicine or tick collars on pets, because pets can run around in the yard, pick up ticks, and come inside and bring them into you in the house. Even if you're not in these exposed areas, they can pick them up in surrounding areas. So after you're outside, it's very important to do a tick check. So there's lots of really great material. I'll feature a lot of it here from the CDC about what you can do for ticks and tick prevention. These are some of those resources, so be sure to check yourself, check children, check pets. Even check your gear and your backpack and your clothing that you might have had out there, because they might be hitchhiking in there and get to you later, as they'll find you over time. It's good to do a tick check in the shower. It's good to shower when you get back in, at least every 24 hours to hopefully avoid pathogen transmission. And even your clothes you can dry on high heat. This will kill any ticks that you might have picked up and are still crawling around on your clothing. So there's some good ways to remove a tick. This is a list of all the ways you should not ever remove a tick. So I'm just going to put these are all myths up first, so don't take this as a suggestion of what to do, but there's many myths out there of you can paint the tick with nail polish and make it black out, or smother with petroleum jelly, or use heat, or burn it with a lighter. And many of these things are not only not very effective, but actually not recommended, because you're more likely to get them to regurgitate into you and kind of have more likely transmission of different things in this process. So do not do these. Not only that they're not effective, they're actually dangerous. So the proper ways to remove a tick: use clean tweezers and grab the tick as close to the skin as possible. So you want to be able to pull all of those mouth parts out. Pull upwards slowly, don't jerk it or twist it, and then clean the bite area as you would any kind of wound. You can dispose of it in alcohol in a sealed container. You can put it on tape. You can flush it down the toilet. If you want to test it for things down the road, it might be good to throw it in some ethanol. You could keep that around. There are different groups that will test it for various things. But it's important, if you do pull it out, it still could attach in a different place. So if you just drop it on the floor, it's still going to be at risk to you. And then after a tick bite, look out for symptoms. Monitor these symptoms for 30 days. And if you have any of these, then reach out to your healthcare provider. If you have a rash, fever, fatigue, headache, muscle pain, joint swelling pain, any of these around after the bite. So in your own backyard, there's some good ways to kind of prevent or reduce your risk of being exposed to ticks. You can create a tick-free zone. So this kind of well-manicured yard is safer against ticks. So you can keep the play areas away from vegetation, remove leaf litter, clear tall grasses, make sure to mow it down. Using wood chips or gravel between lawns and wooded areas might prevent them from making their way in, as you see this kind of perimeter in this picture. You can treat your yard with the acaricides in the spring to reduce ticks, and think about their hosts. So these ticks are going to be in an area when there's many hosts around. A lot of them are on deer and other mammals. So if you discourage deer from visiting your yard, don't feed these mammals and other things that might be out there with these ticks on them, and try to keep deer out of an area. You might have less ticks. So the tick we're going to focus on today is the Lone Star tick. This is widely distributed in the eastern, southeastern, and south central U.S. As an immature, its hosts are medium and large mammals and birds. As an adult, it's medium and large mammals. This is a picture here. Its habitat is usually in wooded areas and forests, and it's known to transmit quite a few things: bourbon virus, heartland virus, bacterial agents responsible for ehrlichiosis, tularemia, stari, and alpha-gal syndrome, as we'll be focusing on here. Here is a great picture from the University of Rhode Island showing the different life stages. So this, you can kind of see the features of the larvae only has 6 legs. So between their larval and nymphal molts, they do gain an extra set of legs. So even if you say this can't be a tick, it looks like an insect with only 6 legs, it could just be a young larval tick. So next I'll pass it over to Dr. Carlson to introduce the alpha-gal syndrome.
Thank you very much. All right, so I want to take a step over to the immunology world that I live in, and just talk about what allergies are and what cross-sensitization is, because alpha-gal is a cross-sensitization. So first off, in terms of what allergies are, surveillance and looking to see if things are penetrating through that might be a parasite or might be something that could transmit parasites to you. When your allergic immune system finds a credible threat, it'll sensitize, it'll activate and form specific components of the immune system to fight off this perceived parasite. Now, certainly in this country, more often than not, the allergic immune system is not helpful. In fact, it's harmful. When that pollen grain lands in your eyes or on your nostrils and you get a sneezing reaction, that's because your immune system made a mistake. It thinks that pollen, when it's releasing its digestive enzymes attempting to fertilize you like it would a flower, it is presuming that it's trying to burrow into you and parasitize you. it definitely is a mistake, and so the reactions that are prompted are unneeded and unhelpful. Beyond unhelpful, they're actually harmful. Now, the immune system, the allergic immune system in particular, can be really quite destructive, and so it tries to be as specific as possible. Any antibody doesn't just recognize a protein as a whole. It actually recognizes a very specific part of a protein called an epitope. And such a narrow target usually ensures specificity of the adaptive immune response. That particular part of that particular protein should make it so that there isn't anything else that looks like that, so that your immune system, if it reacts appropriately or inappropriately, will be a very narrow target that it's looking at. Unfortunately, sometimes that epitope is shared across different targets. A few really well described examples are that albumin from cats. So we all have albumin as a protein in mammals, but our albumin varies going from one species of mammal to another. Cat albumin has an epitope that's very similar to the proteins in pigs. And so there are people who are allergic to cats. They sneeze, get itchy eyes when they're around cats. And when they eat pork, they develop anaphylactic reactions. So that would be an example of an epitope across species resulting in anaphylactic reactions. Not anaphylactic reactions, but even more common than cat-pork syndrome is that birch pollen, this minute particle called BETV1, shares an epitope with apple peels. A protein on apple peels. And so there is a not insubstantial number of people around with birch pollen allergy that when they bite into an apple, if it still has the peel on it, will get a really unpleasant sensation in their mouth. Fortunately, that BETV1 protein is not stable. So by the time it hits the acid in your stomach, that protein will be destroyed. And so it's pretty rare that this cross-reactivity would result in anaphylaxis. There are many other examples. So, for example, if you're allergic to ragweed pollen, there's an epitope in ragweed pollen that's very similar to bananas. And so some people will say, oh, bananas, they're just awful. And other pollen fruit cross-reactivities. So collectively, these are called oral allergy syndrome because the proteins get destroyed by the stomach acid. So most of your symptoms are just in the mouth when you're eating it. And because they're relatively unstable proteins, you can even just put that apple in the microwave for 2 minutes or bake it in the form of an apple pie. And even though the peel is still there, those proteins will be destroyed and you'll be able to eat it. But getting on to the cross-sensitization that we're talking about today is alpha-gal. So there are a number of very unusual things about alpha-gal syndrome, but among them is that it is an example of cross-sensitization. So this alpha-gal, this carbohydrate that's found in the saliva of ticks, is the sensitizing exposure. So credit to the immune system, this really is a parasite. It really is burrowing through the skin. So it is right to be alarmed. It's just unfortunate that the target that it picks in some patients, not most, but in some patients, will be this alpha-gal epitope that is also present on a number of other mammals. Next slide, Mark. So in addition to this unusual epitope being a sugar as opposed to a protein, there are some other really unusual characteristics of alpha-gal syndrome. That is anaphylaxis that occurs when you eat red meat. That is a cross-sensitization. In most patients, the primary sensitization was to tick saliva. Again, the first unusual thing is that it's a sugar rather than a protein. And that may help explain why the duration of the allergy is unusually short. So for most patients, not all, but in most patients, it's a temporary allergy. And after a couple of years of avoidance, avoiding the tick bites particularly, it is common that the reactions fade and ultimately go away. Most other food allergies that you acquire as an adult are permanent. There are kids that get alpha-gal syndrome, but it is more common in adults. The food allergies acquired in childhood, you are more likely to outgrow those. Moving on, again, probably because of it being a sugar rather than a protein, it takes hours after exposure to the red meat to develop anaphylaxis. We believe this is because the sugar, the carbohydrates get incorporated into fat, and fat absorbs more slowly than proteins. This is a theory, so we don't know for sure that it's true, but it's highly probable that this is the case. It does make diagnosis really confusing because maybe you have a really nice meal, you're eating some pork or some beef, and then you go home, you go to sleep for the night, and you wake up at 2 o'clock in the morning having an anaphylactic reaction. Because most food allergy is within minutes, 5 minutes, 15 minutes, 20 minutes from eating, we usually have a really good story that points to what the culprit is. As opposed to alpha-gal, you have anaphylaxis at a random time. The physicians will often ask, what did you do in the hour preceding your reaction? And that misleads a lot of us into targeting something else that is not the actual trigger and excusing the red meat. Red meat is a rare allergy to have, and so it definitely makes the diagnosis more complicated. And then additionally, why it's a complicated diagnosis is the inconsistency of reactions that you have with alpha-gal syndrome. With typical allergies, a person will follow the exact same pattern with each exposure. It may vary slightly with intensity, but usually it is fairly consistent. So if I eat peanuts and I'm allergic to peanuts, and I have hives and wheezing, I'm probably not going to throw up the next time I have a reaction. It will probably be hives and wheezing again. If I'm allergic to milk and I get flushing and vomiting, then in the future reactions, I'm likely to have flushing and vomiting as the pattern of exposure. Again, alpha-gal is not as consistent, and for some patients, it really is just one organ system that seems to get involved, maybe the intestinal symptom, and that the delay in the reaction and the inconsistency of symptoms definitely makes it much, much harder for us to make the diagnosis. In all of this, people who've heard me talk about alpha-gal, I'm often very frustrated about the number of rules that it violates, all of our fundamental paradigms of immunology and allergy that it breaks. One of the editors of one of our allergy journals asked me if I wanted to write an editorial, which was very therapeutic for me to just basically vent about how unusual alpha-gal is. So the reference is up there. Next slide. Another really bizarre thing about alpha-gal syndrome. So again, it's already a little bit strange because it's a cross sensitization. You're sensitized by the tick bite, and then your allergy is to red meat and other mammalian products for reasons that are completely not understood. If you get stung by a paper wasp depicted here, or a yellow jacket, or a honeybee, potentially if you've gotten bitten by chiggers or fleas, and certainly other tick bites, but even in the absence of other tick bites, these other bite and sting reactions seem to bolster the reaction. And that definitely is a lack of specificity. So those paper wasps that can boost the reactions, there is no alpha-gal in there. And so it clearly doesn't make any sense at all. So an additional conundrum that we have to contend with. So when we're trying to get people over the edge so they're no longer reacting, we have to caution them to avoid all tick bites, but in addition, other biting and stinging arthropods. All right, next slide, Mark. So here are our tools for making the diagnosis, and this is, again, some more frustration that we have with this particular diagnosis. The allergy skin tests are not available for alpha-gal, so I can't buy an extract of alpha-gal. I could use pork or other things, but that won't confirm the diagnosis that it's to the sugar, this alpha-gal epitope. So that doesn't exist. Blood tests for alpha-gal do exist, but they are really not specific at all. A lot of us are just walking around with allergic antibodies that are capable of recognizing alpha-gal. Nevertheless, we're able to eat red meat and it doesn't cause us problems at all. And so you would never want to test for food allergy in the absence of symptoms, because a huge number of people, most of the positives, are going to be false positives. You're going to make a diagnosis of a syndrome that you simply don't have. We don't really know why so many people are walking around with these IgE these allergic antibodies that would recognize alpha-gal, and we also don't know why they're not clinically relevant. But so it's a very unreliable test. Then we have 2 exposures. That is, you're going to eat it. Probably the most fun of those is the ice cream challenge. And so you literally just feed somebody ice cream. It is unreliable, so many people with alpha-gal are able to eat dairy and ice cream. The ice cream is good because with the fat, it does hold a lot of the carbohydrates in there, including alpha-gal. So we'll say, yeah, have some ice cream, which is a lovely thing to do in our allergy clinic, but it's unreliable. So you may do fine with it, but that doesn't tell us that you can actually eat the meat that has a much higher concentration of the alpha-gal. It is safe and enjoyable though, so it's not a bad thing to do. When you do react to the dairy, it's usually a milder reaction than you would get with the meat. And so increasingly, we're offering that as the first choice for doing an ingestion challenge. And then only after that goes well would we proceed into the high exposure ingestion. And so for the high exposure ingestions, it really is like have some sausages or some Jimmy Deans or some hot dogs or whatever it is, your mammalian meat of choice, and you just load up on it, eat a bunch of it. Remember that the reactions are delayed. And so very unusual, what we have to do is typically we tell people to eat the food before they come into clinic, knowing that reactions within the first hour are pretty rare. I will say we kind of hold our breath. What if somebody gets stuck behind a train? What if they get in a car wreck on the way in, delaying their arrival in clinic? Nevertheless, nationally, that is our standard. You have them eat it at home and come quickly in, where we can monitor you for 4 hours after the ingestion. The index reactions, it's not unusual for somebody to react 6 or 8 hours after the ingestion, but once you've properly been primed, nearly all of the reactions will be within 4 hours. So we do think it's fairly safe to just block off a big chunk of time. It's very boring for the patients to sit there, but we hope that it's boring. The 4 hours elapse, and then we can release them saying that no, they don't have alpha-gal anymore, or perhaps they never did if it's a diagnostic challenge. Or sometimes it'll end more quickly because they have an anaphylactic reaction. We get out our epinephrine and we treat them so that their anaphylaxis doesn't progress. So again, these are our 4 tests, one of which doesn't exist. The second one, the blood test, is incredibly unreliable. The third one, also unreliable. And the fourth one, which is the gold standard for making the diagnosis or demonstrating cure, it's in our guidelines. That's what we're supposed to do, but it is very dangerous. Next slide. So the additional concerns for our patients once you've made a diagnosis is that we don't know what your threshold for reactions are. So there's a lot of medications out there that contain gelatin, a lot of products that we use in medicine that are derived from pigs, like heart valves, etc. The gel caps are probably the #1 where people get the exposure is very low, and so it doesn't usually cause anaphylaxis, but you can get a lot of intestinal upset. For some patients, not all of them. And the only way to determine if they can tolerate those very low doses is just to give it a try. More dangerous is the stories of these colloid plasma expanders that have alpha-gal in them. So you are in hemorrhagic shock from being in a car accident or a surgery that isn't going well. So you're already in shock, and then you get infused with these colloids that have alpha-gal, and now you have an anaphylactic shock on top of your hemorrhagic shock. So those are very scary. Unfortunately, I've never had a patient that happened with, and there's only a handful of examples, case reports from around the world. Another challenge is that most physicians outside of the field of allergy and immunology have never heard of alpha-gal. Every once in a while, you'll see something hit the popular media about red meat allergy from ticks. It's such a quirky story that the message does get out, but that's usually how physicians hear about it. It's something that's relatively newly recognized, so most physicians wouldn't have had the opportunity to learn about it in medical school, and it's not really a part of the medical school curriculum yet, so even physicians newly trained probably haven't been specifically taught about the quirks of this condition. Allergists have heard about alpha-gal, so we know what it is. But it is such a challenging thing. Most allergists feel uncomfortable with the diagnosis because the tools are so bad, particularly with those ingestion challenges. My clinic here, we're within the hospital itself. And so if we need to call a code, we've got an intensive care unit, we've got all of the support staff that we need. And we specialize in managing anaphylaxis right here in clinic. We carry so much epinephrine. I mean, I give epinephrine every week because we take care of these very, very high-risk patients that we're doing high-risk desensitizations for their drug allergies and things like that. And the protocols don't always go well. So we have a very specialized center. We're equipped for it, but I really don't begrudge my colleagues out in community settings that say, no, this is something way riskier than what I want to handle. But it does mean that most patients don't have access to an allergist that's willing to go through the formal diagnostic process of diagnosing it and then also demonstrating after a couple of years whether or not you've been cured. And then the last really difficult challenge for alpha-gal is that it is rare. We don't even have prevalence estimates. There's no uniform reporting mechanism, so we don't know how common it is. That's especially true because of the variability in symptoms and the difficulty in making the diagnosis. But even if we did really nail it and make the diagnosis every time, it's still probably a relatively rare disease, which makes it very difficult to do research on it. So those are some of the challenges from the physician's standpoint when we approach patients with alpha-gal syndrome. Back to you, Mark.
Thank you. Yeah, and one thing I wanted to follow up with, because this is one of my frustrations, which I'll come back to at the end, but is I just want everyone to be aware of what a mammal is. It's kind of crazy when describing this as a reaction to mammal meat, how many people don't realize what a mammal is. So as we know, a mammal is an animal whose babies are fed with milk from the mother's body, has a backbone, well-developed brain, fur and hair, and is warm-blooded. So all the pictures here are mammals. Things like fish, things like chicken are not. So we'll come back to that, but I just think it's amazed me how much I assumed that the general public knew what a mammal was, and how many times I've had to give a quick biology lesson about what a mammal is. So I had to throw this in here somewhere, it's kind of following up in there. And kind of some of the other interesting research from posted online on the alpha-gal information, which I'll talk about later more, is that we think about the Lone Star tick as the primary vector, but there's other kind of suspected cases or suspected species out there. In the US, there's a few different ticks that are suspected, so a lot of research needing to be done. And then it's also present worldwide in another tick species. So this is bigger than just the Lone Star tick in a small part of the US. This is a bigger thing that's starting to get a lot further. So I think that kind of helps put it into context. So shifting gears, it's going to be a little bit less about facts and figures and a little more about stories now. So I'm going to go through kind of my experience. So as was mentioned, I have alpha-gal syndrome. So I'll go through my background of when I picked this up and progression to today. So on the map, I grew up in Findlay, Ohio. This is well out of the range of the Lone Star tick. I would have been safe if I would have stayed put there the whole time. I went to school for entomology at Ohio State, graduated in 2012. Following there, went down to Disney World and did a really fun internship in entomology. So this is me with Farmer Mickey and catching bugs kind of backstage. You can see the Epcot ball in the background there. Now is when the story gets more relevant to our story here. I went to the University of Arkansas for my master's degree and was there from about 2013 to 2015. While I was there, I did a lot of field work and participated in different things like bio blitzes, where we were out trying to determine how many species we could collect in a 24-hour period. That's what was going on in this picture here. So this picture might be the day I got alpha-gal. I don't know. I can't really track it, but I know I got a lot of tick bites on this day. Following there, I went to Texas A&M University and finished my PhD in 2020, and then finally came here to the city of New Orleans. So that just kind of to understand where I am, and that's some of the complication of the story is that I, wasn't in the area for Lone Star ticks, and alpha-gal very long, went into there, probably contracted it, and then the diagnosis came later. So I'll zoom into those times in Arkansas and in Texas. So when I was in Arkansas, as I mentioned, I was doing field work. So I was out in the field collecting termites. I was out hiking for fun. I can't tell when this might have happened, but anytime I was out in the woods in this part of Northwest Arkansas, I got many, many tick bites. So I started just keeping a pair of tweezers or forceps in the shower. And every time I came back from the field, I would check and usually have to pull at least 1 or 2 ticks off of me. So I got many, many, many bites. This is likely when I contracted alpha-gal syndrome in this time, although it's can't be sure. So I had my initial symptoms in 2015, around the time I was moving to Texas. I know right after I moved there, I went to a family wedding and talked to my aunt, who's a nurse practitioner, about these weird symptoms that I had in the middle of the night that I couldn't really remember if it really had happened or it was kind of a dream or food poisoning, upset stomach. I didn't really know what was happening, but I had some initial symptoms probably in 2015, a couple of years after this continuous spread of tick bites. My first documented reaction was in the fall of 2016, and it was actually I was at school. I was working on my graduate degree, and we had a class project. So we were all in the evening in the lab doing an experiment in the lab, and I started getting these hives all over. So the reaction, this is one of the first questions people ask me. So I wanted to cover this well, is what does the reaction look like? So in this case, I had eaten some meat for lunch. And now it's about 6 or 7 o'clock at night, and we're still in the lab doing this work. And I start getting all these itchy hives. Particularly for me, it was on my joints, between fingers, on hands, ankles, knees, around my ears were very warm and itchy. I had general redness and swelling. So in these reactions, in general, I would particularly get these very swollen hands where my fingers looked like sausages. I couldn't close my fingers. It was often accompanied by kind of lightheadedness and fainting, so I'd have to lay on the ground sometimes. So they're still doing the project on one side of the lab, and I'm just laying on the floor because it's cool on the floor, and I feel like I'm going to pass out. Sometimes I would get kind of a cough when breathing deep, and oftentimes I have kind of diarrhea, upset stomach, the typical symptoms. But at times, if I'd wake up in the night in bed and have to get to the bathroom, I would feel like I'm going to faint and have to stop and lay on the ground halfway there, and it's not a very big distance. And so something, my background in science and in entomology, I don't have a medical background. And so it really took until I went to an allergist to understand that this is anaphylaxis. So I thought that anaphylaxis is like in the movies where your lips swell and you can't breathe and you take an EpiPen. I thought that was the only thing. But as I've learned from Dr. Carlson, it's just the response of multiple organ systems. So as soon as you have multiple systems responding, it's really something to be taken seriously. I didn't realize that because I thought, I've never had a breathing problem. This is just, just a little rash or just a little something happening. So I would usually take Benadryl. It would subside, and with time, my reactions would go away. So I also want to bring up kind of the variability of reactions. So my kind of case that I'm presenting here is my reaction. And as you mentioned, it's very variable across people. There are plenty of other things. So if someone says, I have alpha-gal and this is what I have, believe them. This is not something that they're making up or not something different. Everyone's is very different. I've read and heard a lot about different fume reactions where, if you're in a break room and somebody heats up their lunch in the microwave and it's a mammal product, just the fumes and the smell of that in that building can be enough to send those people into a reaction. As you mentioned, many people can be allergic to dairy or not. I've never had dairy sensitization, so I was able to have dairy the whole time. And mammalian products. So I, again, also had no issue with gelatin or with small amounts of things. If I knew it was there, I tried to avoid it. There's even surprising things that you don't know, that like cornbread mix, you can have to get the vegan version because there's lard in the cornbread mix, the Jiffy cornbread mix. So there's mammalian products everywhere. It's not labeled. It'd be very handy as a patient if they put a little label on there that said not alpha-gal safe, but it's not currently that way. Also, I really want to emphasize the sporadic nature. So they're consistently inconsistent and very sporadic. As I was beginning to get these reactions, the scientist in me said, if I have a reaction today, let's save some of the leftovers of that and eat it a few days later and see if I have a reaction. And all those times I would not have a reaction. So in my mind, I had ruled out that that's what had caused it and was able to move on. So it's very variable and very difficult to figure out. So after that first reaction in kind of the fall of 2016 that I really wrote down, on this day I had this symptom at this time and I started eating this thing at this time of the day. From there till the time I was diagnosed, I had a handful more reactions. So I had at least 6. I had 6 documented, plus probably a couple more in the night that I didn't know. These were to a variety of things, sometimes beef, sometimes pork, sometimes a hamburger, different things. And it came down to finally, at times it was kind of, impacting me and really slowing me down. And so that's when I went to the doctor. I had my final reaction to Ethiopian food in Austin when I was living there and had beef, pork, and lamb. So I had the full trifecta and finally was able to figure out that I had alpha-gal syndrome. But the embarrassing thing is that as an entomologist, I had heard of alpha-gal. I think I could even look back at an email that my mom, who I think is tuned into this webinar as well, sent me an interesting story about, look at this cool bug story. There's alpha-gal syndrome where a tick can sensitize you to meat. And I, knew what alpha-gal was since probably 2012 or 2011, but didn't, was able to rule this out in my head because sometimes I ate meat and sometimes I didn't. And in this time between these symptoms, I was consuming mammal meat freely. So I never limited my mammal meat during these reactions. And it sometimes Like one of the summer of 2018, I went to a conference in Brazil, and we had quite a bit of Brazilian barbecue and different things while I was there. So finally, at the end of October 2018, I was diagnosed. I saw an allergist. I went in and showed him my list of symptoms and foods that I had eaten, and he thought it was a little bit obsessive, and I told him I thought it was alpha-gal, and he said, he's had one patient, because now I'm outside of the area where it's very common. So if you look at the map of where alpha-gal is most common, I picked it up in Arkansas where it's very common, and then moved to Texas where it's very uncommon. So he wanted to test me for a whole handful of things, but he agreed to do the alpha-gal test, and of course everything came back fine except for alpha-gal. And I remember leaving that appointment, and one of the first things I did was I needed some dinner, so I stopped by the grocery store to see what I wanted to pick up, and it really hit me at that moment as I was walking around through the meat area and just realizing all the more things that I couldn't eat anymore. It was a little bit depressing, so it was kind of really hit me as I was walking around the store. And then a couple weeks later, I went to one of my favorite restaurants, that kind of chain restaurant, Piada. It's kind of like Chipotle, but Italian. And so I ordered what I always would get, and it was a steak Piada with pancetta or bacon bits into there. So kind of like a burrito here. So I remembered when I got into line, like, I can't have the steak anymore. Let me get the chicken instead. But I just was an autopilot for I always get, this topping, this topping, this topping, and I still threw the bacon in. So I got home and then opened it up and realized, oh, I had avoided the steak, but still had the bacon. Can't eat this. Because at that point I was trying to be very, very cautious, and even the exposure I was trying to avoid. So it took me a while to adjust into this, and it was difficult to find a lot of information when I was first diagnosed in the end of 2018. One of the best resources I found at the time was this Facebook group, this alpha-gal Facebook group. There's many people in there. Just an example of a post. People are posting, this is my lab test back. My doctor didn't really tell me what to do. What does this mean? And they pose this to other individuals in the group, because there's just not a lot of good resources out there at the time. Now, though, I really want to plug, I found this website through other things, and this alphagalinformation.org has everything in one great place, and is a really, really amazing resource that I wish had been there in 2018. I think I've figured out a lot of these things on my own. But if you go through this website, there's really, really great information, and it's a really great place to find everything all together. But there's tons of alternative foods, so it took me some time to kind of figure this out, but there's alternatives to red meat that are pretty good. Things like emu and ostrich are a pretty good substitute for a steak. In my time with alpha-gal, I've really come to love duck. Duck is a lot like red meat. It's very, you can cook it, undercook it. You can have it more on the rare side, and it's very similar in that same way. So I still really like duck since then. There's also different products that have turkey sausages or things. You just have to be careful, though. And a lot of these things, they're not making this for someone who has alpha-gal. They're making this to be a healthier option, to avoid red meat in your diet. So at times they might have a sausage that's turkey, but it's still in a casing that's pork. So you really have to do your homework. And these things are very difficult to tell. They're not labeled. So when you look at the back, it might say casein, or it might say all sorts of different things. It's not going to very clearly state, although sometimes when it's going for the vegan audience, it will. But in a lot of these things, it's doing this for the health option, not to make it easier. I also had a few Impossible Whoppers, and sometimes eating those, I was worried that maybe they mixed it up, because with all the toppings and for a fast food hamburger, it's not really that noticeably different to go from the Impossible to a regular Whopper. So sometimes I would eat it and say, this tasted pretty good. I hope they gave me the right order today and put the right thing in the packaging. There's also plenty of foods you can still eat. So this is a picture of me at one of our work crawfish boils. I was lucky enough that they made the first batch of crawfish without the sausage in the pot, so that wouldn't be kind of leached into the food. So you can see Barry behind me made me a special batch at the beginning that had no sausage present in there. But you can focus on the great things like seafood. Living in New Orleans, this has been great. You can also focus on poultry. I already mentioned duck, but chicken and turkey are there. You can have fish. So there's tons of options: grains, fruits, vegetables, all sorts of things that you can still eat even with this. The thing that annoyed me the most was just being so picky. I didn't enjoy going to a restaurant and having to ask many detailed questions about, does this have veal broth or beef broth, or what's the base of this, and can I eat this or not, and having to describe it. As I mentioned, many people don't know what a mammal is, so you really have to go through every single option and kind of troubleshoot out what's going on. There's another interesting thing that came along. So the FDA approved these gal-safe pigs. So these gal-safe pigs were created as a way that initially they made these GMO pigs where they took the alpha-gal out of them. And the goal of this was to be using their different organs and things to use these in people and avoid having this reactivity to the alpha-gal in those organ transplants or blood vessels or different things. As a byproduct, they also had all this pork sitting around they didn't know what to do with, so they got it approved for human consumption. And I wrote to this company. They weren't really selling it yet, but they were willing to send out packages. And I got, a cooler full of pork chops and ground pork and bacon that were all gal safe. And in this time, I could still eat these just fine. They had no problem. It's still, you have to kind of get on a wait list. They're not selling it, so it's not as easy to come by. But this is out there and maybe a thing of the future. So as Dr. Carlson mentioned, once I came here, I started doing some of these food allergy challenges with him. So as a timeline here, remember I had first symptoms in 2015, diagnosed in 2018. The next spring, I just wanted to make sure I really had it. So at home, I made myself a steak on the grill. The next summer was took some Benadryl while I was grilling and had a reaction. But then coming back from 2019, I'd spent a couple years without any consumption. So I went to Dr. Carlson and I did one of these taste tests where I came in, as he mentioned, I got to eat at home. So my first goal was to make all the foods that I missed. So I made a lot of bacon, you can see on the top right, and sausage. I really like corned beef. So I made a corned beef roast. I had some sausage, pepperoni, and I made a steak. So I had all of this in the morning and then drove myself down to his office and sure enough had a reaction. So I failed. I had a couple shots of epinephrine and was able to go on that day. I waited a couple years and came back about 2 years later, last October. And then this time I'd found different solutions to find things that were a lot like steak with the emu and duck. So I wasn't excited about that. This time I just wanted bacon. Bacon was the thing that you can't get a nice fatty undercooked piece of bacon in any other way. So this time I just made as much bacon as I could eat and then drove myself in. I got around 1.5 pounds eaten, started to have an upset stomach and then drove in, and sure enough, as I was there, I started getting hives and took epinephrine and was okay and went on with that day. So this led me to kind of wonder, though, about desensitization. So I noticed I had a couple times where I had accidental consumption of mammal products and I had no issue with it. So I had expected, if I ate this, I should have some kind of issue, but nothing happened. And so after that taste test, I began eating half a piece of bacon every day. So just kind of the same way you could eat a peanut a day to get rid of a peanut allergy. We figured this wouldn't hurt. Let's give it a try. As time went on, every couple months, I increased it by another half a piece. So then I was having 1 piece. A couple months later, I was having 1.5 pieces. By the end, I was having 2 pieces of bacon every day. And so after 8 months, I scheduled another taste test. I also got a lunch reservation at a Brazilian steakhouse here. So I could go in, eat lunch at the steakhouse, see how much beef and different meats I could eat, and then go to the food allergy challenge. And this time I passed. So finally, earlier this summer, just a couple months ago now, June 18th, I passed that taste test. So since then, I've been enjoying meat. This is a quick little look at, I was lucky enough to be in Poland last week. And so this is some of the meals, some of the sausages and pork knuckles and meat trays and all the things that now I can eat again. It was really great. We were over to see Taylor Swift. My fiance was very excited to see her in Poland. So while we were there, I had plenty of meat. So this was very good timing that I had passed this test and was now able to eat this meat. So finally, I want to share as a patient, some of the frustrations that I have and kind of a couple stories along the way of these frustrations. So in general, there's just kind of a lack of knowing what alpha-gal is and knowing what mammal products are. So I went to a local drugstore here to get my flu shot one fall and was trying to go in, and I asked the nurse practitioner that was going to administer it if there were any mammal products in the vaccine, if it had, if it had any concern for me. And her first, I said, I have alpha-gal. I was curious if there are any mammal products. And she responded with, what, are you against animal testing? And then I had to kind of step back, explain what alpha-gal was. But by the end, I wasn't really sure that she was really going to be able to read that label and look. Again, at this point, I was still kind of using some of those Facebook groups. So there was kind of general posts that someone would say, I have alpha-gal syndrome. This is my sensitization. I had this flu shot and had no problem. So you could kind of gauge it in the group. Luckily, now there's a lot better resources out there. But at this time, even the medical professional just took it a whole different way. There's also a kind of a pattern of restaurants not taking it very seriously because there's so many people on diets that are choosing what they want to eat. When you come in and say, I can't eat this, I can't eat this, they kind of roll their eyes and say, okay, another, you know, choicefully gluten-free diet person is coming in and making me change everything. And there was a case with the restaurant where I walked through, you know, I can't have mammal, I can't have bacon. Sometimes people don't think it's a mammal, but I walked through every little detail. They served me the food. I was eating the meal and looking at it. I was like, what are these little chunks of meat? And I asked the server and they said, yeah, it's bacon, but it's just for flavoring. It's fine. And I was like, that's still in my food. So it's difficult. And I think that was one of the biggest things that was a frustration to me. I was very okay with finding substitutes and other foods for this. It's probably healthier not to have as much red meat in your diet. But I didn't like having to be this picky person at a restaurant that was, you know, asking them a million questions about what's in there and what I could eat. And then finally, just kind of that in general, the obscurity takes a lot of explanation. So that mammalian products or mammal products is not universally understood. Hopefully with that quick little overview of what a mammal is, everyone here will know. But in general, most people don't know. And even within the medical community, I went for my annual physical this last year, and the nurse practitioner was reading over all of my chart and said, "Oh, you have alpha-gal," and just said, "Yeah." And then we talked about it at the end, and she admitted that she didn't know what it was and was just kind of nodding along at the beginning of the appointment. So even in the medical community, it just isn't out there, and so it's frustrating to have to explain it and kind of sit there and explain what you have to the doctor who's supposed to be taking care of you. So it's been a frustrating road, but luckily, as of just the last couple of months, I've passed out of having the allergy, and now I'm back to a mammal-filled diet. So that's what I had. I'm happy to take any questions for either of us. We can go over that. I also have a QR code to our listserv for the Gulf South Vector grant, so you can scan that QR code or just email cdcvector@nola.gov. And be sure to follow us with the City of New Orleans Mosquito Termite Rodent Control Board at NOLA Mosquito on X, Instagram, and Facebook. We have lots more content like this coming, so be sure to tune in for others. Are there any questions?
Yeah, or I'll take one that's from the chat about the desensitization. So just so everyone knows, the standard is not to try to desensitize any food-induced anaphylaxis. That's the standard. But there's a lot of research now on using desensitizations, and while historically they were incredibly dangerous and people died with the attempts at desensitization, the newer protocols are coming along quite nicely, and so we're able to desensitize more and more food allergy. And there's a couple of different approaches to that. One of them is called a food ladder. That's where you start with a really carefully measured small amount. So peanut, we have pretty good data. It would actually be at most a third of a peanut that you would start with, and that will still trigger a reaction in a lot of people. So you do that first dose in a clinic that can manage an anaphylactic reaction should one occur. So when Mark was interested, like, well, oh no, I didn't outgrow it in my 2 years. It really should have happened. We talked about food ladders, and so there's one international expert on alpha-gal syndrome, Scott Commons in North Carolina, where they have a lot of alpha-gal. I mean, he just sees a ton of patients with alpha-gal and does most of the research on it. And so I told Mark, oh, let me shoot him an email and see what he thinks. And so he said, look, it is a reasonable approach, particularly when you have a PhD level entomologist. This is somebody who really knows what they're doing. Give it a try. Let me know how it goes. So I was able to report back successfully that this is perhaps the first medically endorsed food ladder for desensitization of alpha-gal, but it is not standard. And so most people that you run into, this is not what they would offer for treatment. And then the only other thing I wanted to add is that the variability in where alpha-gal is largely dependent on where the deer are, but it's also dependent on where the fire ants are not. So the reason why New Orleans has very few, in Texas likewise, very few alpha-gal patients is because we have the fire ants eating the larval ticks that then never grow up and are able to bite the people, leading to the sensitization. So very hodgepodge. Different communities can have very high or very low prevalence, and that changes over time as the ecology and people's activities change in an area.
Thank you so much. Dr. Carlson and Dr. Janowiecki. Just any more questions? I think you've addressed almost all of them in the chat. So with that, we really appreciate your really unique perspective on this. And I think it was actually really fascinating from the medical standpoint, from the really entomological, but also from that patient standpoint. So this will be recorded. It's a recorded webinar. We're going to be putting it on our YouTube channel. So the best way to be notified, as Dr. Janowiecki said, is sign up. And we have some other interesting webinars coming as well. All right, everyone. Well, thank you so much. Again, thank you to our speakers for taking their time this afternoon. And thanks for everyone who joined us today. Have a great afternoon.
Thank you.
Thank you all.